In the previous video, we understood the anatomy of gingiva. Now let's take a deeper look at its microscopic structure. Microscopically, gingiva consists of two main components, an overlying stratified squamous epithelium and an underlying connective tissue. These two are connected by the basement membrane. The epithelium is highly cellular, while the connective tissue is less cellular and mainly composed of collagen fibers and ground substance. Now that we know the basic components, let's focus on the outer layer first. The gingival epithelium is the outermost layer that interacts with the oral environment. Based on structure and function, it is divided into three types: oral epithelium, sulcular epithelium, and junctional epithelium. Before we understand these types, let's quickly revise its basic histology. Gingival epithelium is mostly keratinized and has four layers: stratum basale, that has cuboidal cells; stratum spinosum with polyhedral cells; stratum granulosum, that has flattened cells with keratohyalin granules; and stratum corneum, which is outer keratinized layer. Now that we know the layers, let's look at the cells that form them. The gingival epithelium has two types of cells: principal cells known as keratinocytes, and other cells are non-keratinocytes or clear cells. Now these principal cells proliferate by mitosis in basal layer and differentiate into other cells as they migrate towards the surface. Differentiation of these cells involve keratinization. So what exactly happens during this keratinization process? First, the cells become flatter and start accumulating cytokeratins or tonofilaments. Next, the connection becomes stronger through desmosomes and keratohyalin granules are produced in stratum granulosum. As the cells move further upward, they lose nucleus and other cell organelles. Finally, the keratin proteins and lipid accumulates and create a protective layer. Depending on how complete this process is, two patterns can be seen. Complete keratinization leads to orthokeratinized layer with no nuclei in stratum corneum and a well-defined stratum granulosum. While in parakeratinized epithelium, stratum corneum retains pycnotic nuclei and keratin granules are dispersed, and hence granulosum is not distinct. Apart from keratinocytes, there are other important supporting cells as well. These are non-keratinocytes, including melanocytes, Langerhans cells, and Merkel cells. Melanocytes are dendritic cells that are located in basal and spinous layer. They store melanin in organelles called premelanosomes or melanosomes that secrete melanin, which causes pigmentation of gingiva. Langerhans cells contains Burbic granules and are mainly located in upper layer like granulosum. They are phagocytes and present antigens to the lymphocytes. They are found in less number in sulcular epithelium and might not be present in junctional epithelium of healthy gingiva. Merkel cells are located in deeper layer or basal layer. They have specialized neural receptors and are non-dendritic. Now that we understand the cellular composition, let's come back to the three types of gingival epithelium. The oral epithelium covers the outer surface of marginal and attached gingiva. It is about 0.2 to 0.3 millimeter thick and may be keratinized, parakeratinized, or both. Moving inward, we reach the sulcus. The sulcular epithelium lines the gingival sulcus. It is thin, non-keratinized, and lacks retipegs. It extends from the gingival margin to the junctional epithelium and is semi-permeable, allowing bacterial products to pass through. And at the base of this sulcus lies the most important attachment epithelium, junctional epithelium. The junctional epithelium is a band of non-keratinized epithelium that attaches directly to the tooth. It is 3 to 4 layer thick in young individuals but can go up to 10 to 20 layers with age, and it tapers apically to 1 to 2 cells. It attaches to the tooth via internal basal lamina and to connective tissue via external basal lamina. But its importance lies not just in structure but in function. The junctional epithelium forms an epithelial barrier against plaque, and it allows passage of gingival crevicular fluid and has rapid turnover, aiding the repair. Along with gingival fibers, it forms the dentogingival unit. Now the final question: how does this unique epithelium actually forms? After enamel formation, the tooth is covered by reduced enamel epithelium. During eruption, this reduced enamel epithelium fuses with oral epithelium, forming the junctional epithelium. The epithelium then migrates apically and a space is formed, forming gingival sulcus. In a fully erupted tooth, the junctional epithelium maintains attachment to the tooth, and that's how structure, cells, and function come together to form the gingival epithelium. After understanding the epithelium, the next step is to see how it attaches to the underlying connective tissue. This attachment is provided by a structure called the basement membrane, which acts as a link between epithelium and connective tissue. To understand the basement membrane clearly, it is important to first focus on its main component, the basal lamina. The basal lamina is produced by epithelial cells and forms the part of the basement membrane closest to the epithelium. It is extremely thin and can only be seen under an electron microscope. It consists of two layers, lamina lucida, located near the epithelium, and lamina densa, located towards the connective tissue. The lamina lucida helps in attachment of epithelial cells through hemidesmosomes, and the lamina densa provides structural support and strength. Beneath the basal lamina lies the lamina reticularis, which is produced by the connective tissue. Therefore, the basement membrane seen under light microscopy is actually a combination of basal lamina and lamina reticularis. Now let's focus on another major component of gingiva, the connective tissue. It is largely a fibrous tissue and its elements are derived mainly from oral mucosal connective tissue and some fibers like dentogingival fibers that originate from the developing dental follicles. Structurally, the connective tissue is divided into two layers. Papillary layer, which lies just beneath the epithelium and forms connective tissue papillae that projects between epithelial retipegs, and a reticular layer beneath papillary layer. It is denser and is continuous with periosteum of the alveolar bone. Let's focus on connective tissue composition in detail. Connective tissue, also known as lamina propria, has two compartments, cellular compartment and extracellular compartment. The cellular compartment mainly consists of fibroblasts and other cells like mast cells, inflammatory cells, and macrophages. The extracellular compartment consists of fibers and ground substance. The ground substance fills the space between fibers and cells. It is amorphous, highly hydrated, and composed of proteoglycans like hyaluronic acid and chondroitin sulfate and glycoproteins like fibronectin. Now let's focus on fibrous components first. The connective tissue consists of three types of fibers that are collagen fibers, reticular fibers, and elastic fibers. The main collagen fibers that form bulk of lamina propria are of type 1 and provide tensile strength to gingiva. Type 3 fibers that are reticular fibers form supporting network and are continuous with basement membrane and blood vessels. The basement membrane itself has type 4 collagen fibers. The elastic fibers include oxytocin, eulanin, and elastic fibers distributed among collagen fibers. Now, the most important point. These densely packed collagen bundles are anchored into extrinsic fiber cementum just below junctional epithelium. This forms connective tissue attachment, which plays a key role in preventing apical migration of junctional epithelium. Based on their arrangement, these gingival fibers are divided into three groups: gingivodental fibers, circular fibers, and transeptal fibers. The gingivodental fibers are embedded in cementum just below junctional epithelium and are present on facial, lingual, and interproximal surface. On facial and lingual surface, they spread out in a fan-shaped pattern mainly towards marginal gingiva, but some also extend towards periosteum. Interproximally, they extend towards interdental gingiva. Next, the circular fibers. These runs within the connective tissue and encircle the tooth like a ring. They help in maintaining the contour of marginal gingiva. Finally, the transeptal fibers. These are located in interproximal area and run horizontally from cementum of one tooth to the adjacent tooth. They lie between junctional epithelium and interdental bone and are sometimes considered part of periodontal ligament fiber system. So overall, these fibers form a dense network and maintain shape of gingiva, providing tooth-to gingival attachment and connect adjacent teeth and alveolar bone. Now, coming to the cellular elements. The most predominant cell in the gingival connective tissue is the fibroblast. Fibroblasts are present between the collagen fiber bundles and are of mesenchymal origin. Functionally, fibroblasts are responsible for collagen synthesis and production of elastic fibers, glycoproteins, and glycosaminoglycans. They also regulate collagen breakdown through phagocytosis and collagenase activity. An important point here is that fibroblasts show heterogeneity, meaning they are not all identical, and this variation helps in normal function, repair, and response to disease. Apart from fibroblasts, there are other cells present. Mast cells commonly found in gingival connective tissue and are involved in inflammatory responses. Macrophages and histiocytes are part of the mononuclear phagocytic system. They are derived from blood monocytes and are involved in defense and phagocytosis. Now, even in clinically normal gingiva, we can still see small numbers of inflammatory cells. These are plasma cells and lymphocytes, especially near the base of the sulcus, and neutrophils present in relatively higher numbers and seen in both connective tissue and sulcus. So, this shows that even normal gingiva is in a state of constant immune surveillance. Now, coming to the vascular and neural component of gingival connective tissue. The microcirculation, blood vessels, and lymphatics play an important role in drainage of tissue fluid and spread of inflammation. The blood supply of gingiva comes from three main sources. First, supraperiosteal vessels. They are present along the facial and lingual surfaces of alveolar bone and give capillaries that extend towards sulcular epithelium and outer gingival surface. Some branches may also pass into the periodontal ligament. Second are vessels from the periodontal ligament. These arise from the root apex region, extend coronally into gingiva, and anastomose with vessels in the sulcus area. Third are interdental alveolar vessels. They arise from the crest of interdental bone and run parallel to the bone crest and connect with periodontal ligament vessels, gingival capillaries, and supraperiosteal vessels. Now, just beneath the outer epithelium, capillaries form hairpin loop patterns within the connective tissue papillae. These loops helps in efficient nutrient exchange. Along the sulcular epithelium, capillaries form a flat anastomosing plexus running parallel to the tooth surface. In healthy gingiva, the vascular pattern is regular and organized, but in inflammation, it becomes irregular, dilated, and convoluted. The lymphatic system helps in removing excess fluid, clearing debris and microorganisms, and controlling inflammation. Lymphatics from gingiva begin in connective tissue papillae and drain into vessels outside the periosteum, finally reaching regional lymph nodes, mainly the submandibular group. Now, coming to innervation, gingival tissue are richly innervated and most nerve fibers are myelinated. These are closely associated with blood vessels. The nerve supply comes from periodontal ligament nerves and labial, buccal, and palatal nerve. Type of nerve ending present are free nerve endings, in which some extends into epithelium, Meissner's corpuscles for touch, cross end bulbs for temperature, and encapsulated receptors. So, overall, this vascular and neural network ensures nutrition, defense, and sensory function of gingiva. That completes the microscopic features of gingiva. In the next video, we'll move to the clinical aspect, which is very important, and correlate these microscopic features with what we actually see clinically. Thanks for watching. Follow and subscribe to Gamodont for more simplified periodontics.